Singapore and was held since then every 3 or 2 years. It was again planned for this September, as always in Bled, Slovenia. But then as the situation with the new coronavirus worsened, we first decided to cancel the meeting. I have to thank Paula because she put a lot of efforts and energy in motivating the organizing committee to go on with the meeting and to move it to the eSpace. And here we are. So thank you so much for joining us. We have this year 347 participants and registration is still open. And you are coming from 35 countries, which is an absolute record for ICDS. We will start with more simple format today, only with 2 invited lectures. And then each week we will add something new. So we will add poster sessions, we will add the invited short presentations, and of course we will add something that is called sort of chats where 5 or a small number of participants will be randomly mixed in small groups, and then you can, for 5 minutes, discuss to each other. So much for my introduction. And now I would like to introduce our first speaker, which is Rob Fagan. He's a senior lecturer in microbiology at the University of Sheffield. His research career started in Trinity College, Dublin, where he studied surface proteins in extraintestinal E. coli and their role in neonatal meningitis. He then switched to the C. diff surface with a move to Imperial College London, where he worked as a postdoc with Neil Fairweather for 7 years. He established his own lab in Sheffield in 2013 and continues to work on the biogenesis, structure and function of C. diff surface proteins. So, Rob, please, the stage is yours.
Thank you, Maya. Okay, just give me a second, everyone, while we switch around. Yeah, so thank you, Maya. It is an absolutely great pleasure to give the very first talk of ICDS. And thank you so much to everyone who's made it today, particularly those for whom it's either very early in the morning or very late in the evening, or from a few people that I can see on the participant list in the middle of the night even. So thank you very much for coming today. It's a real pity that we can't all be meeting in beautiful Bled. But I'm absolutely delighted that the conference has been able to go ahead online anyway. And thanks again to Maya and to Paula for facilitating that. It is great to be able to talk science even with the current situation. So Maya has asked me to talk about cell surface virulence determinants in C. difficile. Now, it would be very hard for me to talk about the cell surface without at least spending some time on my favorite cell surface structure, which is the S layer. But I have promised Maya that I won't just talk S layer today. So I'll try and give you a brief overview of the state of play in terms of the kind of general roles of C. diff surface proteins in virulence. And I guess the best place to start is really with the question that I guess probably keeps me awake most at night when I think about C. difficile pathogenesis. So as a microbiologist who's interested in host pathogen interactions, I guess we kind of tend to think about this picture on the right being, you know, how bacteria interacting with the host causing damage and living in intimate close association with epithelia and with deeper tissues. But I guess around C. diff, there's a lot of questions as to whether the species is truly adherent, whether it maybe has an invasive part of its lifestyle. And in thinking about these two questions, whether C. difficile likes to colonize things like the mucus layer or whether it's intimately attached to the epithelium, the role of biofilm in all of this and how important that is in vivo, and whether these biofilms are clonal or even mixed species. Now, I think we've made a lot of progress in all of these areas in recent years, but I think it's fairly safe to say, and probably relatively uncontroversial to say, that there's an awful lot we don't know. And I think that we're quite far from a really detailed understanding of the life cycle of C. difficile in the very complex and large ecosystem that is the mammalian gut. So what do we know? Well, we know that C. diff can be isolated from both the lumen and from washed tissue samples. So it clearly has multiple different locations for at least survival in the gut. The relative importance of these different populations in terms of pathogenesis isn't really clear. From very recent work, we know that C. diff can tolerate a little bit of oxygen, which is a good thing, given that although the gut is anaerobic, there is an oxygen gradient from the epithelium out through the mucus layer. So being able to survive in the presence of a small amount of oxygen at least opens up that niche as a potential area for C. diff to live. We know that C. diff can colonize cell surfaces in vitro, and this has been shown by many groups over the years. I've just cited one particular paper here because I love their slightly more complex tissue model in an ushing chamber using both anaerobic and oxygen supplied sides. And very recently, I think this is only last week, we have the first evidence to suggest that spores, at least, can actually get into epithelial cells and that they can act as a reservoir for persistent recurrent infection there. And that's a hugely exciting observation, and it really does open up this idea as to whether there is an invasive aspect to C. difficile survival in the gut. Now, as a kind of a cell surface biologist, my kind of take home from all of this is, well, what do cell surface structures play? What role do they play in all of these processes? So that's what I'll try and touch on a little bit today. Okay, so I guess the best place to start is just to have a quick discussion of the envelope structure of C. diff. So it's a typical gram-positive envelope structure with a single membrane, a very thick peptidoglycan cell wall, which is interspersed with several sugars and polysaccharides that are mixed in among, and in some cases maybe covalently attached to the peptidoglycan. And on the outside, like many bacterial species, C. diff still has a 2D crystalline S layer which coats the surface of the bacterium. Now, since the S layer is kind of my passion, I hope I can be forgiven for occasionally simplifying things down to a model that looks something like this, with just the S layer on the outside. Of course, it's a lot more complicated than that in reality. So C. difficile has flagella, which are anchored in the periplasmic membrane that span the peptidoglycan cell wall and the S layer, and that protrude out into these very large structures on the cell surface. C. difficile also has type 4 pilus, so a slightly more rigid structure that again spans the membrane, cell wall, and S layer, and is responsible for this beautiful colony morphology that we're all very familiar with. It also will have sortase substrates, so proteins that are covalently attached to the peptidoglycan, and an array of other surface proteins, some of which are associated with the S layer and many which aren't. And in many cases, the anchoring of these to the cell surface isn't fully understood. So there's an array of surface proteins on the surface in addition to the S layer. And I think that one of the big outstanding questions at the moment is how all of these structures relate to one another. So how are all these structures associated with the S layer and with one another? I think we're quite far away from actually having a detailed understanding of how that works. Okay, so I'm not going to have time today to go through all of these systems in detail. That just wouldn't be possible in 25 minutes. So what I'm going to do is I'm going to very briefly discuss what I think are the key systems. I apologize in advance to those people whose work of the last decade I've reduced down to a single sentence or slide. I've just highlighted some of the key salient points, and then I've cited some papers. So if you would like to go into this in more detail, hopefully you'll be able to find the relevant information from the groups that really work on these systems. So I'll start off with the flagella. And I'm sure that you're all very familiar with this, but the flagella is an absolutely enormous proteinaceous machine, which is found in the envelopes of the vast majority of bacterial species, C. difficile being no exception. It's anchored into the inner membrane and into the peptidoglycan cell wall. And in the cytoplasm has a series of motor proteins which provide the torsion force to turn a large whip-like filament which is on the cell surface. And it is the movement of this whip on the cell surface that propels bacteria around in swimming motility. So in C. difficile, the flagella is necessary for swimming motility. And indeed, most C. diff strains, at least that I'm aware of, produce flagella and are capable of swimming motility. There are some exceptions. Now, when we look at what the role of the flagella is in virulence, there are some strain-specific differences that have emerged in the literature in recent years. So exactly what role and exactly what exact contribution to virulence the flagella is making is probably up to some debate. What I will say, though, is that in the ribotype O27 lineage, the flagella is definitely very important, and it makes a significant contribution to adherence as well as swimming. And that was shown really beautifully in a series of studies with a motor protein deletion, which produces a flagella, but in which the flagella is not able to move. And that strain is still able to outcompete a strain which has no flagella at all. So a flagellum, even if it doesn't work, is better than no flagellum. Okay, so there's definitely a role in colonization and adherence to the flagella as well. Now, I think what's really interesting, and this is work that's come out of Ria Tomao's lab in the last few years, is that the flagellum is also phase variable. So in a population of cells, we'll find some cells which are producing flagella and some which aren't. And this phenotype is controlled by a genetic switch, so an invertible DNA element, which is upstream of an operon, which includes the genes which encode the proteins for the early assembly of the flagellum. And when that switch is in one orientation, you don't get expression of the flagellum genes, and you are aflagellate. And when it's in the other orientation, you get transcription through the operon of the early genes, and then subsequently the late genes are expressed, and you get the assembly of the full flagellum. Now, I'll talk about this mechanism of gene regulation again later on. But what's really nice about this system is that it's one of the, I guess, first and earliest examples of crosstalk between virulence strategies in C. diff, because within that operon of early flagella genes, there's also SIGD, and SIGD encodes the alternate sigma factor, which in turn is required for expression of the toxin genes. So when the flagellum is phase off, we also don't express toxins. So we're coordinating these two key virulence strategies at a very early stage in transcription. Okay, so the type 4 pilus, again, we've got a structure which is involved in motility, although in this case, it's twitching motility or motility across the surface. And this is made possible by this very rigid pilus structure, which is a polymerized protein filament, which can be extended by polymerization of the filament, so it extends out from the cell surface. It then can grip onto the surface, and then by depolymerization, the pilus can then be retracted back into the cell. And what this means is the bacteria is able to extend an arm, grab hold of the ground, and then pull itself forward. So it's dragging itself along the ground. And I like to think of this like the kind of classic crawlers in a zombie movie. And this is exactly what's going on in these C. difficile colonies when you see this extension of the bacteria out from the dense center, and you get these fibrils of bacterial cells spreading out across the agar. So that's twitching motility across the surface due to this type 4 pili. Now, in C. diff, the type 4 pili is regulated in response to the cyclic diGMP levels in the cell. And this is a really clever way of, I guess, controlling the motile versus sessile switch within the bacterium. So cyclic diGMP acts as this behavioral switch. So in general, populations are either swimming and producing flagella, or they switch to this twitching motility due to cyclic diGMP, where they can twitch across surfaces, but they can also adhere and colonize and form biofilm. And we know that the type 4 pili seems to contribute to adherence, and it seems to play a role in the formation of biofilm as well. And really interestingly, it's been shown that the type 4 pili is necessary for persistence in the mouse gut. So in this experiment shown over here on the right, this is a competitive index experiment where they have a PIL-B mutant, which is this extension ATPase over here. So a knockout in the PIL-B gene competed against a wild-type strain. And if they were equally fit in the guts, you would see that everything clustered around this dotted line, which is 1. But actually what happens is the PIL-B mutant is very dramatically outcompeted in the gut. Okay, so you need type 4 pilus to be fully competent as a C. diff in that competitive in vivo environment. Okay, and then the last of these non-S layer surface protein families that I want to talk about are the sortase substrates. Now, in many gram-positives, these are by far the most common mechanism of surface protein display. So, for example, Staph aureus really relies on this mechanism for coating its surface with protein. And basically, a sortase substrate is covalently attached to the peptidoglycan. So this is physically attached to the peptidoglycan cell wall. And this linkage is catalyzed by a family of enzymes called the sortases. And C. difficile seems to have only one functional sortase, which is SRTB. And what this protein does is it recognizes a motif at the C-terminal end of the protein, which is classically LPXTG, but it will tolerate variations to that sequence. And it cleaves after the threonine and creates an amide bond to the free amino group on meso-DAP in the cell wall in this peptide crosslink in the peptidoglycan. Okay, so the protein then is physically attached to the cell wall. In C. diff, and I hope I haven't missed any recent papers here, but at least 5 putative adhesins have been identified as bona fide sortase substrates, so these 5 proteins here. At least one of these has had its adhesive properties confirmed, and this is CD2831, which has a collagen hug domain which is exposed on the cell surface. And this protein, well, in isolation, this domain is able to bind to type 1 collagen. Okay, so this looks like it's definitely adhesive. And what's really nice about these sortase substrates is that there's an extra mechanism which also ties these adhesive proteins back into that cyclic diGMP switch that I talked about between flagella and type 4 pilus, so swimming and twitching motility. Because in the presence of low cyclic diGMP, a gene which encodes the protein called PPEP1 or ZMP1 is expressed. But just starting from the inside out, SLPA is translated as normal on your ribosome, and then it is post-translationally secreted across the membrane via SEC-YEG. And the energy for this is provided by a dedicated motor protein, ATPase SEC-A2. So it has its own SEC-ATPase, which is kind of important for this very high throughput secretion system that's needed for the S layer. Once the protein is on the outside of the cell, or at least outside the membrane, it actually undergoes a cleavage event, which is catalyzed by a cell surface protease called CWP84. And that yields 2 fragments, which we call the low molecular weight and high molecular weight S layer proteins, or SLPs. These 2 SLPs go on to form a very high affinity heterodimer. And it is this heterodimer that is actually the basic subunit that then goes to self-assemble and form this 2D crystal that coats the cell surface. And this whole crystal is then held in place by interactions between the individual proteins and a sugar, a polysaccharide of the cell wall called PS2, which appears to be universally found in the cell walls of all C. difficile strains. So that's how you put together, in very general terms, an S layer. But as I said, the S layer is not just SLPA. SLPA, in fact, is just one of 29 members of the cell wall protein family. These proteins are all characterized by having 3 copies of a cell wall binding motif. This is a PFAM04122 motif. And this is the part of the protein that interacts with PS2 and anchors the protein onto the cell surface non-covalently. Okay, so all of the members of the family have these 3 copies of the cell wall binding motif. And in general, most of them have other bits to the protein, which in many cases we can bioinformatically predict functions for these. But actually, very few of the proteins have experimentally confirmed functions. And I'm just going to talk you through a few of those now and let you know exactly what they're doing in terms of the pathogenesis of infection. So the first one that I'll talk about is CWPV, and this is by far the largest member of the CWP family. It has its cell wall binding domains at the N terminus, and at the C terminus, it has a number of repeating protein elements. So these are protein repeats. These are variable in number, so different strains will have different numbers of repeats, and they can also vary dramatically in sequence from one strain to another. Okay, so they vary both in sequence and in number of repeats. But what's really interesting about CWPV is that this protein is phase variable, and it's expressed by only a small minority of cells in a normal mixed population. So what we have here is an immunofluorescence image, which is showing the S layer, SLPA, labeled in red, and CWPV labeled in green. And what you can see is that only a few cells in the population are expressing CWPV under these conditions. And typically it's between 1 and 5% of cells, okay? So this is a phase variable system. And Maya, actually, what you may not know is that it was the mechanism of the phase variation of this protein was actually discovered over beers at the second ICDS, which was in Maribor many years back. There was a few of us talking about Jenny Emerson's data and looking at her beautiful phase variation pictures. And we were talking about the possible mechanisms that could explain this strange observation that Jenny had. And actually on the plane back from Maribor, she was flying back to London on her own, and sitting there with her computer, she actually discovered the genetic switch that controls CWPV by eye in the DNA sequence. And if anyone's ever looked at these kinds of things, you'll realize what a monumental task that must have been to find inverted repeats by eye in a genetic sequence. It's crazy. But there you go. A connection to ICDS going back many years. So as I mentioned, the phase variation is controlled by a genetic switch. So this is very similar to the flagella story that I told you earlier. So the upstream of the CWPV gene, there's this pair of inverted repeats, and the sequence of DNA in between can undergo very rapid and high frequency inversion from in both orientations. Okay, so that it can basically be flicked like a light switch from one orientation to another. And in one orientation, CWPV is expressed, and in the other orientation, it's not. Okay, so it's a clear on-off switch. Now, this is a bit of a puzzle because the promoter for CWPV is actually upstream of the switch. So how does the switch control expression? And Jenny had a very nice solution for this, in that she proposed that in one orientation, a very strong stem loop structure could form over one of these inverted repeats, which is shown here at the bottom. And this acts as a transcriptional terminator to stop transcription at that inverted repeat. Whereas in the other orientation, a much less energy favorable stem loop is formed, and the transcriptional apparatus is able to plow through this, and so CWPV is expressed. So that's the model for control of CWPV. And I should say that the inversion of this switch is actually controlled by a specific recombinase, which is RECV, which mediates the switching of the orientation of the switch. And that's actually the same recombinase which also controls the flagella switch, although they appear to be regulated independently. I think, well, that's my understanding anyway. Rita may correct me on that. Okay, so that's the regulation of CWPV. So we have these, and it's mediate interactions between cells and aggregation, which maybe contributes to biofilm formation. Okay, so that's CWPV. I can just quickly scan through a few of the others. So we'll protein family. We have CWP66, which was one of the first to be characterized. And back in 2001, it was shown that pure CWP66 fragments or antibodies to CWP66 could block adhesions. This is pure protein, either N or C terminus of CWP66. An antibody to the N or C terminus could block adhesion of C. difficile to vero cells. So CWP66 seems to have a role in adherence, at least to this cell type. CWP84, which I told you already was necessary for maturation of the S layer, has another role. It has a very potent proteolytic degradation activity against some extracellular matrix proteins, including fibronectin. You can see the degradation here, laminin, and vitronectin. Okay, so this protein may be involved in the tissue damage that we associate with very severe C. diff infection. And the final one that I mentioned is CWP2. And again, this protein doesn't have a lot of homology to anything in the databases other than SLPA. But a CWP2 mutant is significantly less able to adhere to Keiko2 cells. So it seems that this protein may also be an adhesion. So lots and lots of adhesins. Okay, so what about SLPA itself? Well, when this was first characterized by Neil Fairweather's group back at the very start of the century, so it was Emmanuel Calabi did the early work, Emmanuel showed that the high molecular weight SLP, but not the low, could adhere to these human biopsy, these are colon biopsy samples. And then a few years later, it was shown by Gertrude Vidansen's group that purified SLPs or antibodies to either the high molecular weight or low molecular weight SLP could block adherence to Keiko2 cells. And these were the BBE brush border variants of Keiko2 cells directly play a role in adhesion to tissues. So that was, I guess, where we were kind of limited in working with pure proteins because it was very difficult to isolate an S-layer mutants. We eventually managed this about 5 years ago. And we were very excited at the time because we were very quickly able to show that the S-layer mutants that we had isolated was avirulent in hamsters. So this is the Kaplan-Meier survival curves for hamsters infected with C. diff. And you'll see that those animals that are infected with a wild type, this is R2-9-1, succumb very quickly to infection. Our S-layer mutant doesn't kill hamsters at all. In fact, it doesn't cause any disease symptoms whatsoever. And a complemented mutant kills the hamsters again. Okay, so we've got a very clear role for SLPA in virulence. But what was really puzzling was that when we looked at what was coming out of the hamsters in terms of their feces, but also in their cecal contents after euthanasia, we couldn't see any difference in the level of colonization between those animals that succumbed to wild-type infection and those animals infected with our SLR mutants that were showing no disease symptoms. So we had normal colonization, but no disease. Now, at the time, trying to attribute that to the SLR directly was a bit problematic because the SLR mutants had pleiotropic phenotypes and also didn't produce normal levels of toxin. So we couldn't separate out these two phenotypes. Now, more recently, we have solved the structure of SLPA. I'm not going to have time to talk about that today. Hopefully, you'll have heard Paola or myself talk about this before. But suffice to say that we're now able to make much more targeted changes to the SLR to really dissect its function in more detail. So the first one of these that I'm just going to tell you about is a deletion of the second domain of the low molecular weight SLP. So we completely delete this domain, which is normally exposed on the cell surface, and you end up with a smaller low molecular weight SLP, which we call delta D2. Now, interestingly, the strain that's producing this truncated protein still forms a normal SLR. So it looks like a perfectly normal SLR, just lacking one of the surface details due to this lacking domain. Okay. But what's interesting is that this strain causes significantly less severe disease in mice. Okay, so this is mice now. Mice infected with a wild-type R289-1 rapidly lose weight over 2 days and then gradually recover their weight as they clear the infection. Those animals that are not infected continue to gain weight throughout the course of the infection. And when they're infected with our mutants, which is just lacking this one domain of SLPA, they still lose weight, but a lot less weight than the wild-type infection. And they also recover towards the end. So we get much, much less severe disease. I'm not going to show you all the details, but also have lower pathology, less severe pathology in the guts of these animals. And crucially, these animals are colonized to very similar levels. So these are those infected with wild type and with our domain 2 mutant, colonized over 24, 48, and 72 hours of infection. The colonization looks very similar. And critically, the mutant produces as much toxin as the wild type, so this is not statistically significantly different. This is wild type toxin levels and the mutant toxin levels at 48 hours and 96 hours post-infection. And you can confirm this by Western blot. So these animals have less severe disease, but they're producing just as much toxin and they're colonized to very, very similar levels. Okay, so I'll just leave it there with some, just sum up with a few points. So I hope in that very brief overview I've been able to get across to you that C. diff is a very flexible pathogen with a very wide range of surface proteins that contribute to disease. Swimming and twitching motility are likely key in certain circumstances, but the picture is very complicated by strain-to-strain differences. We have many proteins that can contribute to attachments to tissues. We have a surface that's dominated by an S-layer, which is decorated with CWPs to add functionality. And crucially, we've now been able to show that the S-layer is required for full disease severity and that it's not just about the toxins anymore in C. diff infection. And so I will stop there. It's gone a couple of minutes over, sorry. Just quickly to thank the members of my group in Sheffield, past and present, and my very good collaborators, Per Below and Roy Chowdhury in Sheffield, and the other members of the Bug Slayers Consortium in Glasgow, Jill Dowse and her group, in Newcastle, Paolo Salgado and her group, and of course our funders, the BBSRC, MRC, and Wellcome Trust. And thank you very much for your attention.
Thank you very much, Rob, for this extremely nice overview. What we are missing now in Zoom is this function of applauding and applause, and you know. So, but yeah, there are little hands that are showing this. So, yes. But I would just like to say again that if you would like to join the discussion or if you would like to ask something, we originally thought that it would be best to put your question in the chat or just to put in the chat, you know, like your name or so. Currently, I don't see any questions from the audience. Just, oh, there is, yes, Secretariat MM. Please go ahead with a question. Oh, hi, VIP class. Please
Maya,
unmute yourself.
Thanks for organizing this great talk, Rob. I'm wondering about the pleiotropic effect you see in a SLPA mutant, and whether you have done any characterization about what is causing this, since the lack of toxins might hint at a regulatory event, right? So maybe if you would characterize the mutant, you might get a handle on why it produces less toxins.
Yeah, so unfortunately, that's a very difficult question to answer. I don't have a satisfactory answer for you. We don't really understand all of these effects yet. So in addition to the toxin production defect, the strain also doesn't sporulate properly. And I have a sneaking suspicion those two things may be connected. We've not found any genetic changes that could be responsible. So I think you're right, it is going to be regulatory. We're still waiting to do the RNA-seq experiment that I think might shed some light on this. I think this is probably, though, not an unfamiliar story from a lot of different bacteria. When you change things dramatically in the envelope, that can have a lot of downstream effects in the cell that are very hard to predict. And in many cases, working out the exact causative links can be really challenging. So I know that's not really answered your question.
Good. Thanks.
I talked a lot anyway. I don't know if that's got value, but yeah, it would have been a bit of a short answer. We just said I don't know.
Yeah. So is there any other question? Otherwise, I would just like to say hello again to everybody who joined us during Rob's talk. It's there is a Rob, somebody has sent you copy directly in the chat. Can you look at the chat?
Yeah, hang on, sorry. I'm going to stop sharing actually because
okay, yeah.
I can't see everything. Okay, so Vince has asked me if when we delete the outermost axial or longitudinal distribution of the organisms, do you mean in terms of the actual systems of where it is in the intestine, Vince?
Oh, could you just unmute yourself, Vince, and ask?
Bob, can you hear me? No?
Yes, can do.
Yeah. So quickly, sorry. Actually, yeah, because this gets to the idea, is it adherent or not? So do you see it, you do fish, do you see it in the lumen more? Do you see it on the side more? Or is it in different regions of the gut, like moves from the cecum to the colon, et cetera, et cetera?
So, sorry, yes, I completely fair. Okay, so cecum colonization seems to be pretty much identical. So we see pretty much equal levels of colonization, both in terms of tissue and lumen-associated organisms. So we don't get any change in the cecum. In the colon, it's a little different. I'm trying to remember exactly what the difference was. There was a slight difference in terms of the number of spores that were present. We also quantified vegetative cells and spores, lumen and tissue associated. I believe that fewer spores, the domestication mutants in colon. Now, whether that's due to transit and things being washed out, or whether it's a difference in sporulation efficiency in that environment, I don't know. We've looked at sporulation in vitro, and there's no difference. If anything, actually, it sporulates slightly better in vitro, very slightly. And I think that's just a fitness effect because it's not having to produce this massive amount of protein. But I mean, from what we're able to do with the relatively finely dissected out intestines, we can't see any dramatic differences in terms of where the organism is living, which was a bit of a surprise.
Yeah. Okay, thanks.
Okay.
Yeah.
So there is one more question, which will be also the last one. Rob, can you see it in the chat? What about non-toxigenic isolates in terms of the outermost domain? Are there any naturally occurring strains?
So I'm not sure that there's a definite correlation. You can find non-toxigenic strains with multiple different S-layer types, but there are at least 13 different sequence types of S-layer that have been characterized to date. At least 13, and that's mostly coming from European and US isolates. And some of those strains are non-toxigenic. So there seems to be a wide distribution of non-toxigenic types with S-layer types. One of the S-layer types is naturally lacking what we would normally consider domain 2. But that strain of C. diff, so that's the, I'm trying to remember now the details. I think Neil Fairweather's on here, might correct me. It's S-layer cassette type 11, which is the only one that's glycosylated. Produces a truncated low molecular weight SLP, which looks very much like our deletion mutant here. And as far as I know, that strain is still virulent, or at least it was isolated from a human infection. So that's a toxigenic strain. So kind of in a roundabout way, we don't see a correlation between S-layer type and toxigenic status. And the only strains that I'm aware of that are naturally lacking this domain are kind of weird anyway, because they've got this strange glycosylation effect with the whole surface covered in a glycan. Yeah.
Thanks, Rob, a lot. So now we are moving to our next speaker, which will be Christina Lanzas. She's associate professor of infectious diseases at North Carolina State University. And before joining this university, she was assistant professor of epidemiology at University of Tennessee and senior personnel at the National Institute for Mathematical and Biological Synthesis. Her research interests are the epidemiology of healthcare-associated infections and antimicrobial resistance. Her lab combines data, epidemiological analysis, and mathematical and computational approaches to study pathogen dynamics and assessment of control strategies. One of the focus areas of the lab is the transmission and control of C. diff. So we are now moving a little bit from the research side to more clinical side. Christina, please.
Thank you. I'm going to share my screen. Let's see. Okay. Do you see my PowerPoint? Yes. Perfect. Thank you. Thank you very much for the invitation. It would have been lovely to be in Slovenia, but definitely it's great to see that so many people are coming online. So as mentioned, this is going to be a bit more applied topic. And the way I approach this presentation is pretty much assuming that a lot of you probably are very unfamiliar with the topic of computational modeling and infectious disease modeling. So what I'm aiming today, as I was putting this talk together, is actually give a very, hopefully, non-technical introduction. And so I'm hoping that, people get out of this talk an idea about how these models look like, what are some of the purposes and uses of these models. And then I'm going to give a little bit more my perspective about where we think we have accomplished so far on C. difficile modeling and what we still have a lot of work to do. So to give a little bit of perspective about these models, indeed, the use of infectious disease modeling is actually incredibly old. And I'm referring here to the use of what we often define as mechanistic models. So these are models that we are really aiming to provide mechanistic understanding, so trying to really understand the processes that goes into transmission. And so the first models really come down to the 19th century. So the first model mathematical approach to understand disease transmission really dates back to the 1889. And a lot of the sort of foundation of the methods, it really evolved on the first part of the 20th century, perhaps some of the most relevant work is the work that Ross conducted to understand malaria transmission. And in fact, the model that he developed, we still use today to really sort of provide a framework to understand transmission of malaria as well as other vector-borne diseases. So after that kind of first phase, infectious disease modeling kind of went quiet for a long time until kind of had another new phase in the 90s and 2000s. And the main reason is that pretty much, infectious disease models came to use again as we really face new challenges with infectious disease, particularly first HIV, but then all the emerging new infectious disease that we are having along the way. Some interesting landmarks, it was, for example, in 2001 was the first time that actually models were used to provide real-time guidance. So maybe some of you remember the foot and mouth disease outbreak that happened in the UK. This is an animal disease, but it's hugely transmissible and it costs a lot of money. And that was the first time that they actually used the models to try to figure out what was the best approach to the control of the disease. And there was pretty much a lot of analysis, what we call a post-mortem, about whether this worked, whether the models were right, they were wrong. But since then, pretty much they have become a part of public guidance. And definitely with COVID-19, you are seeing they really, with no other data, another way of going, basically that was for a while the only information that, we were able to generate through the models. And I'm sure there's going to be also a big postmortem about what, worked well for COVID-19 or not. So when we look at infectious disease, and we look a little bit more close to healthcare-associated infectious disease, we see pretty much the same trend. We start to use these models pretty much along the way when we start to have a new awareness about the really burden and cost of healthcare-associated infections. And some of the highlights about what things we have been doing a better job on understanding through models has been particularly the understanding, for example, of healthcare workers and the role that they have in transmitting disease, providing an understanding about the dynamics of hand washing and why they are so important and why sometimes they work and others not. There's been a lot of work also trying to understand the interactions that the different healthcare settings are involved, and so how relevant, considering not just as a single hospital, but the interaction between different hospitals and different healthcares. And more recently, there's been more assessments of interventions and looking at aspects of such as cost effectiveness. And when we look at CDVC, it kind of mirrors the same thing. Really, surprisingly, we have been only doing modeling for really a decade at most. And pretty much it's been very limited until probably the last maybe 3 or 4 years. And so it does mirror the same. A lot of the early work that we performed was really to understand some aspects of the dynamics of disease and the transmission. And more recently, there has been a bit more focus in trying to understand what control strategies are and how we are able to deploy those more easily. So in the context then of C. difficile and healthcare infections, what are some of the applications of these models? One and foremost is that transmission is something incredibly difficult to study, basically because transmission is something that we don't observe directly. And so oftentimes we may use these models to try to measure things that are hard to measure, such as colonization rates, contribution of different transmission pathways. They definitely, they have been used extensively for forecasting, so providing projections of burden of disease, number of cases, and so on. And probably, perhaps, the areas that they can contribute the most, perhaps not yet, but probably is one of the areas that we're going to see the most growth, is really providing what we call in silico testing or simulation, so we can actually evaluate interventions. And that becomes very useful in situations where, first of all, sometimes we don't have other ways to understand the contribution of different interventions. If you think about things like bundles, where basically you are combining a certain number of interventions, but we really don't understand which one is the one that is really contributing the most, or whether there is an interaction among those interventions. And the other particularly useful point when it comes to compare interventions is that basically we often don't need to wait until we have data, and sometimes they can actually help to precisely design or help the design of those interventions. And so one of the things that we see now, for example, there is a flare of models trying to assess things like vaccination, for example, which, it has not been implemented in place, but definitely the models are helping to think what are some of the factors that we need to consider as we think about deploying these new interventions. And one area that maybe sometimes is not quite considered, but it does help, is that oftentimes if we combine data from the clinical studies into the models can help as a way to expand what we call the external validity of the studies. So basically the ability of use the data of a clinical study that has been done in a certain population and trying to see what will happen if you were to apply this a slightly different population, a slightly different conditions, and so on. So in general, models are a great way of doing really what if case scenarios in all sorts of ways. I'm not going to get too much into details about how they look like, but I really want to emphasize a couple of things. First, that they really all have a common framework. So the way we study transmission is by first thinking about the disease from the point of view of the infection, not just the clinical status. So we really need to understand who is being infected at what time. And so oftentimes what we see is like we're always going to be starting thinking about the disease by classifying the population, in this case, the patient population, into discrete categories based on where they are in the infection status. So oftentimes we use what we call these SIR frameworks. So we may consider susceptible, so those individuals that have not been exposed yet to the disease. If somebody gets infected, oftentimes we're going to have a phase that we call exposed, where individual is infected but not infectious, so it's still not a risk to other individuals in the population, and eventually individuals become infectious, where they are actually now a risk for the rest of the population. And for most disease, there's going to be a phase where the infection is not more transmissible, either because somebody acquired immunity or basically death. So when we look at C. difficile, how this framework look like? Well, first of all, it kind of varies. So we have a few models with slightly different combinations of what categories or what states we may want to consider, but basically there are some commonalities. So this is a nice review from Gingras in 2016, where he kind of summarized some of the natural history that it's used on these models. And so some of the things that we see pretty quickly is that there is an agreement about the fact that basically as infectious categories comes, you really need to consider both the asymptomatic colonized patients as well as the CDA patients, so those that are symptomatic, and that's, as you see, common from most frameworks. In some cases, we may consider additional categories. For example, the fact that not everybody is susceptible to the disease. And so we may consider that we want to capture the transition from what we call sometimes resistant patients. So those are the ones who have an intact microbiome. And so we assume that basically they have colonization resistance, and therefore they will not initially be susceptible to C. difficile. And in some other instances, particularly when really we are trying to attempt to capture transmission pathways, we'll see often also consider the role of the healthcare workers. And actually we use the same model that we use for malaria transmission, basically assuming that in addition to our patient's status, we will have also the status of the healthcare workers as a vector of disease. And so those healthcare workers, they can be either colonized or uncolonized. So despite the variability on the models, the other point that we really need to address on these models is precisely how individuals go from that susceptible category to an infection category. And so Oftentimes, as again, the models may vary in the way we address this, but oftentimes the risk of colonization is going to be some function of the patient's susceptibility. Again, whether we consider that, antibiotics, for example, may play a role. What are the potential sources of transmission? And so when we think about C. difficile, right, as we was mentioning before, we often really explicitly account both what is clinically infected individuals, asymptomatic colonized individuals, as well as sometimes we may want to explicitly account for the healthcare workers and room contamination. And when it comes to the mechanistic description of transmission, that can vary greatly, but one of the probably trends that we are seeing recently is that we are getting more and more access to data that is collected through sensors. And so oftentimes we will see a pretty detailed description about the interactions between patients and healthcare that comes with using sensor data. Another point I want to make, just to finish sort of this very quick introduction about these models, is that oftentimes they come in two flavors. One is that what we often define as compartmental models. Those are basically models that we use the compartments that we mentioned before to divide the population. So if I'm in a healthcare system, I can divide patients based on whether they are susceptible, infected, but the point is I don't know who belongs in which. Basically, I don't know the identity of these individuals, I just know my average. To contrast to that, we have seen also most of the more recent models are what we call Asian-based models. So those, very briefly, are models that they consider each individual. So now I know what is the status of each individual in my population. Difference between these two, basically, these kind of models, the compartmental models, oftentimes we can describe them by equations. Here is just one equation just to give you an idea how these things look like. But the idea is that they are a little bit more transparent, and so we can do things like really analysis on these models to understand their dynamics. Asian-based models, they are great because you can add a lot of details to the models. Basically, they are, in some ways, we can think about these models as really almost like video games, because basically we're sort of attempting to recreate the reality in a certain way. So they are all code. I mean, these things is not a mathematical model, it's basically a code-based model. So they can add a lot of detail, but at the same time, it means that they are also far more complex, more difficult to analyze, and definitely they are what we call data hungry. So you need a lot of data sources to fit on these models. And oftentimes we use, for example, a lot of electronic record data to really fit these kind of models. Some people will favor one way models over the other ones. In reality, like everything, they have strengths and weaknesses, and really it all depends what kind of data that you have available, what is the objective of the model. So personally, I don't really consider one type of model better than others, but definitely it's all about what you need for the objective that you have in hand. So when we think about then these models and the application in clinical research, one of the questions that, as I'm myself reviewing what we have done on modeling is really what kind of evidence we are bringing to the table. And so oftentimes, when we think about, for example, clinical research and epidemiological studies that we use to assess evidence, oftentimes you probably have seen something similar to this, what we call the pyramid of evidence, where basically even before looking at a specific study, just knowing what kind of a study design you already have an idea about what is the strength of evidence that type of study brings to the table, basically because, some types of studies have more risk of bias or internal validity being in question. And so we often favor randomized controlled trials over things like case control. And so when it comes to models, it's like, do we have anything similar to this? Oops, sorry. Basically, not quite, but this is my take on it. If we are to be using models for decision making, you may want to always look models that they're actually using data real world data, and so they are calibrated in one way or another. It will be giving a little bit more strength to the internal validity of the model if, in addition to the calibration, they provide also internal analysis. So you have things like a parameter sensitivity that gives you an idea about how the outcomes of the model change as you change parameters. Oops, going backwards. And it will be great if oftentimes these models are not only have a data that has been used to calibrate, but actually use additional data sets that were not involved in the calibration to evaluate these models. I will point out, though, that oftentimes where CDVCL and in general healthcare models are, we tend to be on this realm. You see very few models that they have been actually validated with additional data sets. And the same as pyramid here, at the end of the day, how we built understanding from these models is that you want to have enough models, the way that basically you can actually see the whole body of models, not just one model. And so you have different models that they have been built with different assumptions, that they have been built with different data sets from different populations, and they all maybe give you more or less the same consistent results, let's say for an intervention, then you feel more confident that actually probably, that results are valid, and they probably are valid more across populations. Again, this is still a little bit challenging to do, but hopefully that's where we are. Again, one of the challenges that we have is really we need way more models, and we need more tested and more published studies to really be able to get to that point. So when we look back then again about CDVCL and some of the things that we have been doing on the last years, a little bit, it mirrors what I said before. So a lot of the early studies are studies that were done with compartmental models. And here I'm just like giving you one of the examples from my early work. Those initial models were really aiming to provide a little bit of general understanding about the dynamics. And what we are seeing more recent times is more agent-based looking at interventions. So some of the things that we have learned from the transmission dynamics from these models is one that really, which seems to be very much the case with a lot of healthcare-associated infections, not only C. difficile, that oftentimes, a hospital is not an island in itself. And within hospital transmission, often it's not sufficient by itself to really keep these pathogens endemic. And so how we know that is when you use these models to estimate something that we call the basic reproduction number. And this is probably a measure that, it's been on the news with COVID-19 a lot, and maybe people now are very familiar. But basically what it tells you is that if you were to include, let's say, in one infectious individual in a hospital that otherwise everybody is susceptible, whether that individual will be able to cause more than one infection, and if one individual is able to cause more than one infection, then of course you can have an outbreak take on or get the disease become endemic. And so what we see for C. difficile is that basically a large portion, about 50% of occasions where, and this is looking at very broad range of parameters, that's not the case. One factor that influences a lot whether an individual then will contribute to new infections really is the length of a stay. So how long an infectious patient is in the hospital. So if the within hospital transmission, then it's not enough to maintain transmission. What is maintaining transmission? And so one of the things that we have seen on the last 5 years that they have come to be is models that not only take into account a single hospital, but they really look at transfer of patients within the healthcare networks. And so healthcare networks here are represented from a study that look at healthcare networks within the state of California. Basically what you see is that in this case, the visualization that you see here is that each of these nodes represent a healthcare facility, and these lines represent the connections between the nodes, which are based on transfer of patients. And very quickly, we realized that actually there is a very dynamic healthcare networks with a lot of transfer of patients. And when you basically compare the information that you get from this network data to C. difficile rates, one of the things that we tend to see is that in general, if you have a facility that is bringing in transfers, and so it has what we call an in-degree, so bringing transfers from another facilities, oftentimes those are associated with high C. difficile infection rates. And one thing that we have come to understand, I think, fairly well, and generally for all, again, healthcare pathogens, not only C. difficile, and here I give you an example for CRIs, is that a lot of the interventions needs to be happening really at the coordinate healthcare network rather than in single facilities. And so simulations models are telling us that really, if you were to have some sort of coordinate approach among facilities that are highly connected, oftentimes you will see a greater impact than just having individual efforts by a single unit. What else we have get from the models? As I mentioned early on, all the models basically, they really acknowledge explicitly that both asymptomatic colonized patients for C. difficile as well as CDI patients contribute to disease. That's also something that has been shown, particularly for studies that use genomic data, or they do some sort of tracing, where oftentimes we see that really a small percentage of the CDI patients can really track back to prior CDI patients. So we know that there are other sources of transmission. And we also know that the symptomatic colonization oftentimes can be particularly high. So what we know from models is that oftentimes they are acknowledged. I have found particularly difficult sometimes to pull the data about the contribution of both relative contribution of asymptomatic colonized patients versus CDI patients. But oftentimes those studies, the ones that they report, they can go about very close, like 50% contributions. Another indirect evidence about the role of asymptomatic patients come also from the studies that they have been testing potentially interventions that focus on asymptomatic colonized patients. And here again is another case, as I was mentioning before, that is something that actually is not recommended in general on the clinical guidelines, but nonetheless, from modeling point of view, we can really evaluate and test and see what we are finding. And so this is from a model that we put together really just to particularly address this question about the potential role of asymptomatic colonized and what will be the effects of actually testing and applying contact precautions early on. And what we see is that when we look at broadly range of colonizations at admission, so really looking from 5 very conservative percentage to really high percentage, and also looking at different test characteristics in terms of sensitivity and turnaround time, things that we see is that for CDI rates, those that are acquired to the hospital, we often see changes from 10 to 25%. So basically applying interventions to those patients will provide an additional control point. Again, we learn more when we see different studies looking at the same thing with different point views. This is a different study looking at the same question. We came up about relatively similar results. So screening and contact precautions for asymptomatic colonized gave about an additional 20% reduction on the CDI cases. To point out that when those were combined antimicrobial stewardship, they really projected a very high decrease on CDI rates. Another point then of interest that lately, as I mentioned, is like most recent model work is being to try to attempt to assess interventions with agent-based models. And again, the idea behind using agent-based models is that you can add a lot of details. So for example, we have been building models that they have a very detailed antibiotic exposure history for the patients. Other models that have a lot of detail about the interaction between healthcare workers and patients. And so these are, as I was saying, fairly data-hungry models. And so where we are there is that it is a bit difficult to compare results of different studies, basically because they oftentimes the same intervention may be implemented as slightly different with different levels of degree. And the other element is that oftentimes we quite not see the same interventions apply across the study. So just to give you here an example for 4 studies, so I'm not reporting changes on CDI, but just provide you the ranking of how different models evaluate different interventions. And pretty quickly we see that rankings vary hugely. Here we have 2 studies ranking daily cleaning as one of the most effective interventions that they test. In other cases, for example, these other 2 studies, antibiotic stewardship was ranked among top. And sometimes in this case, basically, there are interventions that basically you don't have them across a study, so you don't have much to compare with. So, as I was mentioning, really we're very early on the studies. So interventions, comparisons, we really have insufficient studies to provide really a robust picture of what interventions may work from a modeling point of view. And reason is that we are varying interventions, implementations, and I haven't mentioned bundles, but bundles are equally difficult to compare. Reporting guidelines, it will be great to apply, and so a little bit like we have done with clinical studies and epidemiological studies, we need similar type of reporting guidelines for models for evaluating interventions. And something that has become quite useful, I think, from COVID-19, particularly the fact that sometimes we have, for example, CDC puts together pandemic planning scenarios by which everybody who has a model is suggested that evaluates the same type of scenarios. That would be great if we had something like that for models, so we can compare the same interventions across models, so we know how to benchmark these models. So just very quickly to finish, we still have a very few published infectious disease models. They probably are more out there that are being used at healthcare level, but maybe not published. And so we're still probably, if we have to build evidence about interventions based on these models, we have to have a really more models to look at this. But overall, the models have provide what I would say interesting information about things like connectivity among healthcare facilities, the role of asymptomatic colonized patients, so aspects of the dynamics in general we lag behind when we are looking at evaluation of control strategies, and we definitely need a way to benchmark the results. And last but not least, I just want to thank to everybody who over the years has been involved in collaborating with CDFICIL, from my lab members to other folks at NCSU, Tennessee, and Washington University. And of course, as always, things are not possible without the funding, in our case by NIH and CDC. And that's all what I have for today, so thanks.
Thank you very much. It was very nice, and I think we now, have better picture how the models are actually working. So thanks for putting this in such a simple and effective picture. And yes, I would like to invite our participants now for questions. And as Paula said before in the chat, you just unmute yourself and ask the question. Yes. And while we are waiting, I was looking at your one of the initial slides where you were showing this R number.
Yep.
That exactly that maybe this one is also showing, and you were saying that this is within hospital transmissions are also sort of influenced by the length of stay. And I was thinking that maybe this is actually showing us that the part of the increase in the community CDI is basically due to this spill out of the hospitals, in addition to all the community reservoirs that are now so modern. So are you modeling also the community infections?
So we have looked a little bit on the community, but you are right in that one of the things that you see is like, and I think one of the trends that you see with the surveillance data is that you have increase on the attribution that of the CDFICIL that is hospital acquired community onset. And some of that is a trend of increase the length of stay in the hospital, have become infected in the hospital. Now they are getting infected when they go back to either, their own households, or whether they go back, particularly for patients that are highly susceptible, back to nursing homes or long-term care facilities. So definitely, and that's why there's been a little bit more awareness of maybe not just modeling hospital level, but thinking about broadly about the community and broadly about the networks because of these interactions that are happening. On the community side, is always a big challenge in the sense that oftentimes what the dynamics that you see at community is always very slow growing, just because of the size of the population. You see a lot of changes when you're only having looking at the population of 1,000 beds in a hospital versus when you're looking at millions in. So those dynamics always very, very slow, and oftentimes not maybe well captured on these models.
Yeah. And where do you see the future of modeling? And how quickly do you think it will really get into even more extensive use than they already are?
So we are seeing a lot of neat applications happening because of we have way more data available. I mean, we are all on a data overlooks, and so we start to see a lot of applications that integrate more. I wouldn't say necessarily big data, but a lot of data sources with these models. And so we'll see a lot of growth on that arena. But I do think that if we ought to take some of the results seriously from these models, we really need to do a better job of being able to compare models, which right now we are not quite there, particularly, as I was mentioning, complex models. They are so complex that basically you have a model here, another model here. It's very difficult to really provide an understanding of what things are in common, what things are different. And so we really, as I say, kind of, for me, resembles what we have done in epidemiology. We really need more reporting guidelines. We need more consensus about how we are putting these models together and how we are reporting, so we understand what we are getting.
Okay. So, is there any other question from the audience? Don't forget to unmute yourself. I don't see anything in the chat now, so, well, in this case, thank you, Christina. And this brings us also to sort of the end of today's session. And I would just like to, again, thank you all who, to all our participants of ICDS. And even if we are not, at the venue, and we don't have all these social events, which, by the way, we are trying somehow to also implement in this virtual space. There are still some costs associated with the organization, and therefore I have to thank our sponsors. And you see some of them, and others are listed in our webpage. Before we say goodbye and good night, I would just like to remind all poster presenters, so please send us your posters so that we can put them on the website. I have to apologize because I misspelled the email address in my announcement. But we will send a corrected announcement, and you can find the correct email address of the Secretariat also on our web page. And with this, Paula, if you have some further housekeeping note, otherwise...
Yes, sorry. It's taking me a while to unmute myself.
Okay.
No, I just wanted to say a few things regarding what we're planning in terms of the posters. So as Maya has said, we'll have them on the website as soon as people submit them and we put them up. But we're also hoping that in each session we'll start highlighting a few posters and we'll contact the posters presenters for those details. But we're also planning to have a forum for people to ask questions to the poster presenters. So the posters that we are highlighting in a particular session, the presenters for those posters will be available on Slack for discussions, and we will send out information on how to join those Slack channels and how you can interact with the presenters there. We're also hoping to do that for the speakers for the sessions as well. So if there are any things that don't get covered in our live discussions, you can follow it up on Slack. So that's kind of all I wanted to say. Thank you very much. And if there were any technical problems, we apologize, and we'll do our best to improve for next time.
Yes, and I just thank you, Paula. I just got to the question from Stu. What happened to Mark Wilcox's talk? Yes, one of the disadvantages of these virtual meetings is that the speakers can get unavailable because you don't have them on site. And this is exactly what happened to Mark. Mark is heavily involved in COVID diagnostics. So he just had another appointments, and his talk was rescheduled. And I'm sure this is he will not be the only speaker, and this will not happen only once. So we will keep surprising you. Thank you very much for joining us today, and see you in a week time. Bye-bye, have a nice day, and good night.